Not all proteins are required at all times, as it is inefficient for cells to transcribe and translate all its genes all the time. Cells must regulate gene expression depending on the environmental conditions. An example of gene regulation is the lac operon of E. coli. The lac operon is a group of genes that encode proteins which allow E. coli to use lactose as an energy source. E. coli bacteria uses glucose as an energy source, although lactose can be broken down for energy as well. Since glucose requires less time and energy to use, it is a more efficient energy source. Therefore, only if glucose is unavailable and lactose is available, E. coli bacteria will use lactose. The bacteria will then express the lac operon genes, which encode enzymes used for lactose metabolism. The lac operon consists of a set of structural genes, an operator site, and a regulator gene. The structural genes encode enzymes that metabolize lactose. The genes are transcribed as LAC-Z, LAC-Y, and LAC-A. The LAC-Z gene encodes an enzyme called beta-galactosidase, which splits lactose into glucose for energy. The LAC-Y gene encodes a membrane protein called lactose permease, which helps bring lactose into the cell. And finally, the LAC-A gene encodes an enzyme called transacetylase. The operator site is a DNA sequence that regulates transcription of the structural genes. The regulator gene, also known as LAC-L, which encodes a protein called a repressor. These genes are transcribed so the LAC repressor is always present. The LAC operon also contains a number of regulatory DNA sequences where specific proteins can bind. First, the promoter, which is the binding site for RNA polymerase, which is the enzyme that performs transcription. Next, the operator is a negative regulatory site where the LAC repressor binds to. The regulators turn the operon on and off in response to lactose and glucose levels. This is a LAC repressor, lactose sensor, and the catabolite activator protein, or CAP, the glucose sensor. The LAC repressor takes cue from the environment about the concentration of lactose within the cell and regulates the production of lactose metabolizing proteins by binding to the operator and inhibits transcription. When glucose levels are low, CAP binds to CAP binding site and helps RNA polymerase attach to the promoter, driving high levels of transcription. Scenario 1. The absence of lactose for E. coli cellular metabolism. The LAC repressor is active and binds tightly to the operator, preventing transcription by RNA polymerase. It physically blocks RNA polymerase, which prevents transcription. Scenario 2. The presence of lactose for E. coli cellular metabolism. With the presence of lactose, the LAC repressor loses its ability to bind to DNA. This alteration occurs from a small molecule called allolactose. Allolactose is an example of an allosteric inducer. It is a small molecule triggering an expression of a gene operon. The LAC operon is considered an inducible operon because it has the ability to turn off and on. Once the repressor is inactive, RNA polymerase binds to the promoter and transcribes. When lactose is present, some lactose molecules transform to allolactose within the cell. Allolactose binds to the LAC repressor and causes a conformational change. Thus, it can no longer bind to DNA's operator site. While the operator free of obstruction, RNA polymerase is able to bind to the promoter region. Transcription of LAC genes begin.